Okay, good afternoon everyone. Good afternoon, good afternoon. Good afternoon Shivani, good afternoon Priya, good afternoon Ananya. Good afternoon everyone. So friends, please listen to me very carefully. Good afternoon Neha. Okay, good afternoon everyone. So please listen. See, we really don't know when the exam is going to happen, but according to my knowledge, I feel strongly the exam will be on September 3rd week. Okay, that I am feeling. Good afternoon everyone. Please continue. The exam will be on September 3rd week, what I feel. Okay, but I am not saying 100% the exam is going to happen in the month of September 3rd week only, but what I am pretending that exam will be in the month of September 3rd week. So friends, today date is August. What is that today date? It is August 8th. So what I do is I will take the classes till the September 10th. So August 8th to September 10th I will be taking the classes. What I'll do friends, in this particular series I will just cover 45 topics, but please believe me strongly, out of this 45 topics I can give you the great assurance that 23 to 25 topics very surely comes even in the part C as well as the part B also. Okay, so good afternoon all. So just imagine friends, even though if you get 20 topics, I am telling you, I am giving you the guarantee of 90 to 100%. Good afternoon everyone, good afternoon, please continue. I am giving you the guarantee of 90 to 100% that out of 45 topics that what we are discussing friends, very surely you will get 23 to 25 topics. So and one more thing, you all people know that good afternoon all. In case of a part CSIR, in part C, you all people know that we have 75 questions and we have to answer 25 questions. So what is the guarantee that I am giving to all of you? So out of this 45 topics, I am 1000% sure you can get 20 part C questions. Okay. So if you correctly answer, I am telling minimum, I am using the word minimum. Okay. 20 to 480 marks will be in your hand. Okay, so if you get 80 marks, undoubtedly, if you do a little effort in the part B and part A, 100% you are going to qualify the exam. So my only request to all of you is follow this particular 45 topics very thoroughly. Okay, very thoroughly. What you have to do, once I complete the topic, what you do is keep the PPT with you. Okay, if you have the time, you go for the any kind of a reference book, but right now we don't have a much amount of time. So what you do friends, just do the PPT and just read the Pathfinder once, enough. Okay, just read the PPT once and read the Pathfinder once, and you no need to worry about that. For every topic in the class itself, in the session itself, I will cover the part B and part C questions in the topic itself. Okay, good afternoon all. In the topic itself I will cover the part B and part C questions, so you no need to worry about it. What I want you people to do is, as soon as I complete the lesson, keep the PPT with you. If you have the time, just refer the standard reference book of only one that particular topic. Otherwise, if you don't have the time, at least read the Pathfinder once. That's more than sufficient to understand this. Okay, now I have chosen 45 to 50 topics. From which units? Did I selected only few units? No. So friends, I have analyzed this 45 to 50 topics from all the 13 units. Okay, from all the 13 units I will be taking minimum 3 and maximum 4 topics. So what do you mean by the word minimum 3 and 4? Based on what I have analyzed all these topics, based on previous 8 to 10 question papers, which the question in which the questions asked very oftenly. Good afternoon Saima. So what I am requesting to all of you, finally I'll just summarize and we will go to the questions, sorry, lesson. In this particular till when I will take the classes? Till September 10th. I will cover 45 to 50 topics from all the 13 units, and I am giving you 100% assurance that out of this particular 45 to 50 topics, undoubtedly you will get in the part C section 20 questions minimum. I am using the word minimum only, okay. You may get a 20 to 23 also, okay, as well as you can get at least 10 to 15 questions in the part B as well, okay. So overall, if you read this 45 to 50 topics that what I am going to tell you currently, you can get more than 100 marks in the upcoming CSIR September exam. Now you can ask me one question, sir, that sir, till now I did not started my preparation. If I really follow this 45 days, will I crack the exam? I never say yes and I never say no, okay. See, this 45 to 50 topics will be useful, and it will be very highly useful for the students who already doing their preparation from last 6 months or from last 8 months. For them it will be more effective because you already read it. Now I revise and I will explain with respect to the question wise. As a result, what is happening, it will boost your confidence levels. If you did not study anything so far, and if you want to crack within this 45 days, I never say that you will qualify, and I also never say that you may not qualify or may qualify. So you people can give the try, but the people who are studying from the last 4 to 6 months or 8 months, undoubtedly these 45 topics are going to help you to secure more than 100 marks from all 13 units, okay. So now we are starting with the unit 2 first. So I'm just reminding to all of you what are the various topics that I'm going to take in this particular unit 2. Cell division and cell cycle regulation. You all people know that if you look into the 22 February paper, 2020 paper, and if you consider the 2019-18, from the last 10 question papers, minimum 8 times the question was appeared from the cell cycle and regulation. Then friends, the second topic that I am going to take from this particular unit is the transposons, okay. And the third topic that I am going to take from this particular unit is the microbial physiology, where you can expect numerical as well as the graphical questions. And the fourth topic that I am going to take is, friends, organelles and its function. Since I have already covered the organelles once, what I will do, friends, I will, in this particular fourth session I am talking about, since I already covered, what I do is, instead of teaching you the organelle and its functions, what I do is I will directly solve the part B and part C questions which were appeared from the organelles and its function. As a result, anyway, we have already solved this concept very nicely. We will only focus on this, but this I will teach the concept as well as part B and part C question. And whatever the 50 concepts that I am teaching, you no need to worry. I will only solve every part B question and every part C question. The only thing you have to do is revise, okay. And for every topic, the PPT itself will help you up to 90%, okay. Now let's start. Cell cycle and its regulation. Friends, all the students, listen to me. What kind of questions that you can expect in this cell cycle regulation? There are many ways are there you can expect the question. You can get the question from graphical, you can get the question from the experimental based, as well as, friends, you can also get the question statement based, okay. Only these are all the three ways that you can expect the question from this particular cell division and its regulation, okay. Now I am not explaining you the division, I am explaining you the regulation, because 99.99%, friends, we never expect the question from the division in our CSIR exam. What we expect the question is from the regulation. And if you look into the 2022 February paper, what kind of question we got, friends? We got only one question, that is the experimental based question. If you look into the 2020 paper, we got the question from the factual statement, but we got the question. So that's why you have to know very depth of this particular concept. Okay, now let's start. Okay, now please do not interrupt me. First, you people listen to me for 20 minutes continuously with a great patience. Then, friends, automatically I'll solve the questions. You can get a crystal clear concept. Okay. My ultimate goal is I want you to get 100% of the concept, okay, and I want you to get the confidence in this particular topic to crack that particular question at one attempt. That's all my goal. Okay, now let's start. So let me explain you for 20 minutes. cell cycle and checkpoints. Friends, let me give you one simple word. Why we require the checkpoints? Now the question is very clear. Without a checkpoint, don't you think the cell cycle will be continued? What I can say is no. Without regulation, the cell cycle never ever be continued. In one word, you know what I can use the word, general word for the checkpoint, I can use the word toll gate. Okay, you know, right, when we are traveling in the highways, compulsory our vehicles will be stopped at the toll gate. Once the checking is over, then only we will move, otherwise we never move. Same way, whenever the cell is getting divided, the cell has to check compulsory at each and every phase. It has to check at G1 end of the G1 phase, it has to check at the end of the G2 phase, even it has to check at the M phase also. So the checkpoints are basically, friends, acting as the surveillance mechanism. Now you can ask me a valid question that, sir, what happens if the checkpoints are not checking? What happens if the checkpoints get mutated? I can tell you one common point, friends. If the checkpoints get mutated, which leads to a major instability, which leads to the genetic instability. You all people know one common point. Most of the birth defects, most of the genetic birth defects that are actually occurring because of the genetic instability only. That genetic instability occurred because of the mutations or because of the failure of the checkpoints. That is the reason, friends, it is very important and it is very mandatory that every cell at every checkpoint, it has to be checked. Once it qualifies that particular checkpoint, then only it can move and perform its function. Okay, now in one single line I can tell you that checkpoint is basically decide what it will decide, the cell has to move to the next stage or not, in a simple word. So it will decide whether the cell has to move or not for the next round of division. Very simple point it is. Continue everyone. And how many checkpoints are there? There is one. First, there are total 3 major checkpoints are there, friends. The first checkpoint, we will be calling it as the G1 checkpoint. Now if you ask me a question, sir, what exactly happening in the G1 checkpoint? At the G1 checkpoint, the cell will check. See, we all people know very well, G1 phase is the preparatory phase for the S phase. Friends, in the S phase, what is happening? DNA replication is happening. If the DNA replication wants to happen, lot of enzymes has to be synthesized, lot of ATP are required. So where all is getting prepared? Undoubtedly, friends, they are all getting prepared at the G1 phase. So in single word, the G1 checkpoint is going to monitor all the events that are happening in the G1 phase. If the cell is not properly prepared in the G1 phase, it is impossible, friends. The checkpoint never ever allow the cell to enter into the S phase. Okay, now you all people know that in the S phase, what is happening, friends? DNA replication is happening. Now the question is very clear. Is there any checkpoint in the S phase? No, there is no checkpoint in the S phase. There is a checkpoint at the, good evening everyone. There is a checkpoint at the end of the G2 phase. See, friends, I called that G1 phase is the preparatory phase for the S phase. Similarly, I will say G2 phase is the preparatory phase for the M phase. So what is happening in the M phase, friends? Obviously, in the M phase, the cell is getting divided. But friends, if the cell wants to be divided, it requires lot of protein, lot of ATP, lot of other essential enzymes also required. Now where all are getting prepared? Undoubtedly, friends, they are getting prepared in the G2 phase. If the cell prepared completely whatever they require, then only, friends, the cell is going to enter into the G2 phase, M phase, otherwise it never enter. And so first checkpoint is at the end of G1 phase, second checkpoint is at the end of the G2 phase, where the third checkpoint is present. Third checkpoint is present exactly at the in between meta phase to the ana phase. Okay, if you ask me the single function performed by this particular metaphase to anaphase checkpoint, I can give you the answer that it is majorly involved in checking. What it is going to check? It is going to check whether the spindle fiber formation occurred properly or not. Spindle fiber formation occurred properly or not, that can check at this particular checkpoint. That's why friends, the M phase checkpoint, you can also call it as friends spindle checkpoint. Please never ever confuse it with this particular point. So be patient friends, I want you to answer all my questions. If you are this 45 days, every day 1.5 hours, I am expecting only one thing, that is your patience and interest. First is most important is interest, followed by your patience, and my responsibility to help you to secure more than 100 marks. Okay, just focus, because they are all very broad concepts, you have to know very depth. Now all the students look into the picture friends. There is one G1 phase, there is S phase, there is G2 phase, and there is M phase. Let me quickly answer my questions. So end of the G1 phase, there is one checkpoint, you can see the picture. End of the G2 phase, there is one checkpoint. End of the in the M phase, especially at the meta phase, there is another checkpoint. Now the question is very clear. Now you can ask me a valid question. Sir, in the S phase what is happening? The DNA is getting replicated. Now you will all get one doubt that, sir, how do we know whether the replication occurred accurately or not, because there is a no checkpoint here? That's a valid question that you are asking. But the point I want you to understand, in the S phase only friends, there are a lot of proteins are there. That proteins are going to control this particular replication in the S phase, especially friends MCM and CDK. These two, I will explain you friends, don't worry. These two are the proteins which are going to control this particular S phase. Don't worry about that. So now we are going to explain one by one checkpoint very briefly and very elaboratively. Okay, first listen. Okay, now when I use a word here, I have said it to you that friends, every phase can be regulated by a different checkpoints. Now what actually the checkpoint is made up of, I have used a word. What does the checkpoint is made up of? As I am repeatedly saying that every phase is under the control of a checkpoint. What does the checkpoint is composed of? Everyone keep it in your mind friends. The checkpoint is generally composed of a CDK molecule as well as the cyclin molecule. These two molecules friends, or these two substances or these two proteins are going to control the entire cell cycle and entire checkpoints. Now most of the students will think that only one type of CDK and only one type of cyclin is going to control all the phases of the cell cycle, which is absolutely wrong statement. They can say that in a given 4-marker question friends, as one of the statement they can raise the question that what they can raise the question, similar CDK and cyclin molecules are controlling the entire cell cycle, which is absolutely wrong statement. In the G1 checkpoint, different CDKs and different cyclins. In the M phase, different CDKs and different cyclins. In the end of the G2, different CDKs and different cyclins. Not the same type of CDKs and cyclins are going to control the cell cycle or the checkpoint. Now all the students listen to me very carefully. This is a very important already asked part B question as well as the part C question also. So pay attention. There are two proteins are there. One we call it a cyclin protein, another protein is called as a CDK protein. Okay, now CDK protein, we all know that cyclin dependent protein kinase. You always remember as CDK. Now let me tell you one interesting point about the CDK. What is that interesting point? CDK having a long lifespan. CDK having a long lifespan, whereas friends cyclin having the short lifespan. So what is the statement I would like to conclude here? That is cyclin, everyone listen, cyclin will synthesize us, cyclin will activate the CDK, and cyclin will degrade. That's all. Because cyclin won't withstand for the longer time. Cyclin synthesizes, major function of the cyclin is activating the CDK, then cyclin automatically get degraded. Now, how many types of cyclins are present in each and every cell? All the students remember, friends, we are having total 4 cyclins, that is called G1 cyclin, G1 to S phase cyclin, S cyclin, and friends M cyclin. Total how many are there, friends? Total 4 types of cyclins are present. That's what I have given you a statement. Friends, only one type of cyclin never ever control all phases of the cell cycle. That's not possible even. Different cyclins are going to control the different phases. Now listen up beautiful statements here. Very important statements. As I have clearly told you that CDK and cyclin together controlling the cell cycle. That is what the statement I have given. But let me tell you one interesting statement. This particular CDK, which is an important protein involved in the checkpoint, regulated by 2 ways. Always remember, CDK is regulated in a 2 ways. One is the CDK can be regulated by the cyclin molecule also. Second point, friends, the CDK can also be regulated by the mechanism of phosphorylation and dephosphorylation. Now, what is the valid statement every student has to remember? Suppose example, if the CDK molecule comes and attaches to the cyclin, then I never use the word that CDK molecule is active. I never use. In other words, CDK molecule only phosphorylated, then I never use the word that CDK molecule is active. When I can use the word CDK molecule is active, I can use the word CDK molecule is active whenever it is associated with the cyclin also, whenever it is also whenever it is phosphorylated also. Then only I can use the word CDK molecule is active. Whenever the CDK molecule is active, then only it can control the cell cycle. Otherwise, it never control. Anyway, let me tell you. Friends, what is the function of a CDK molecule? Name itself indicating na kinase. What is the function of a kinase? Adding the phosphate group. That's all. Adding the phosphate group to the targeted protein. But let me tell you one interesting point. This is really interesting point. Everyone focus. Remember, I said that suppose example, this is a targeted protein. This is a target protein. If this target protein wants to be phosphorylated, CDK molecule will just add the phosphate group. Now what I am saying is, first how does the CDK get activated? If the CDK, suppose this is the CDK molecule, okay? Now, what is the goal? If the CDK molecule wants to be activated, it should be added by the cyclin. And second mechanism, what did I say? It also has to be added by the phosphate group. Now, you have to get a valid question that, sir, where exactly the phosphate group associated to this particular CDK molecule? In the CDK, there are in the active site of the CDK, there are 2 tyrosine amino acids are present. Every student has to remember compulsory the name of the amino acid. In this, there are 2 tyrosine amino acids are present, friends. Among this, 1 tyrosine amino acid, listen everyone, very good. 1 tyrosine amino acid, if it get phosphorylated, then we can call it as stimulatory tyrosine. 1 tyrosine can act as a stimulatory, and if another tyrosine get phosphorylated, we can call it, my friends, inhibitory tyrosine. 2 tyrosines are there. If one get phosphorylated, CDK will become stimulated. If another get phosphorylated, CDK will get inhibited. Now you have to get one more valid question. Sir, then how do we know where the phosphate group is adding? Does the phosphate group is adding to the stimulatory tyrosine, or does the phosphate group is adding to the inhibitory tyrosine? Sir, how do we know that? That's a valid question, right? That you have to understand upon binding with the cyclin. Once the CDK molecule associated with the cyclin, keep it in your mind every time, only the tyrosine molecule, which is a stimulatory in nature, that only get phosphorylated. Very simple point. If CDK molecule is not associated with the cyclin, then undoubtedly what is happening, the tyrosine, which is a type of inhibitory tyrosine, that will only get phosphorylated. Very simple point. I am explaining you in the form of a picture. Don't worry at all. I will explain in the upcoming slides with the picture. Don't worry. Now you have to answer a small question. Now listen. We have discussed very clearly that different cyclins are present in the different phases of cell cycle. At the same time, friends, different CDKs are present in the different cycles or different phases. Now please keep it in your mind, friends. This kind of questions are extremely important for the part B. So it is very important and mandatory. You have to remember them. Here you no need to use your brain. Here you have to remember. Yeast, we know that some yeast can undergo the reproductive method as a fission. Some yeast can undergo the method of budding. Whichever the yeast can undergoing the method of fission, there is only one cyclin molecule and only one CDK is present for the entire cell cycle. Whereas the yeast which is undergoing the mechanism of budding, one CDK will be present, and friends, more than one cyclin will be present. There will be one CDK and more than one cyclin. That's okay, fine. If you don't remember the number of CDKs and number of cyclins present in the yeast, there is no problem at all. What you have to remember is, my friends, in case of our body, vertebrates, that is very important. You have to remember, which is already asked in the exam, and there are great chances are there that we can also expect the similar kind of a question in the upcoming exam also. So more than one CDK interact, G1 cyclin, G1 to S cyclin, S cyclin, M cyclin. In very simple words, with the G1 cyclin, G1 CDK will be interacted. With G1 to S cyclin, G1 to S CDK will be interacted. With the S cyclin, S CDK will be involved. With the M cyclin, M CDK will be involved. And what's the unique statement you have to remember? Don't think that all the CDKs are doing the same function, because the CDKs at different phases, they will perform different functions. Now this is the table. I want every student has to remember compulsory, which you can expect in the part B question, which you can also expect as one of the statement in the part C. Even friends, in the ICMR JRF exam also you can get this kind of questions from the cell cycle regulation. So friends, please remember that without fail. Okay, now listen all of you very carefully. So in this table, what was written? The type of CDKs written, the type of cyclins were written, and they are controlling what stage of the cell cycle it has written. So it is very important. See here, there is no need to use brain, you remember it. Listen to me, friends. CDK4 and CDK6, these two under the control of cyclin D. What do you mean by that? If the CDK4 and CDK6 wants to be activated, you will see in the next slide, they have to be phosphorylated compulsory, and most importantly, friends, they have to be associated with the cyclin D. What you people has to remember for the exam point of view is which CDK controlled by the which cyclin and what is their major function. So cyclin D is, good evening, continue Deepika. Cyclin D is going to control early G1 phase. You have to listen my statement. I am using a word, early G1 phase. What do you mean by that? You all know that in the G1 phase, everything is getting prepared. Correct, na? So whatever the proteins that are getting prepared, that entire protein synthesis under the control of cyclin D. That is what I want you to remember. Then friends, when you look into the CDK2, it is under the control of what, friends? Cyclin E. And please listen, this is the one which always makes the confusion. Here, cyclin E, friends, it is not at all controlling the entire S phase. That is absolutely wrong statement. What is the correct statement is that, friends, cyclin E just initiating, just starting the S phase. That's all. It is just starting the S phase. You know, right? In the S phase, replication has to start. So that phase just started by the cyclin E. Whereas cyclin A, which is controlling the CDK2 and CDK1, required for the regulation of the S phase also, friends, as well as completion of the S phase also. And CDK1, which is regulated by the cyclin B, it is the one which control the M phase, that is the division phase. Now all the students remember one of my statement. You people listen. CDK2, listen friends, this is important statement. CDK2 alone controlled by cyclin E. CDK1 alone controlled by the cyclin B. But if you look into the third one, CDK2 and CDK1 together under the control of the cyclin A. There is a great difference. Most of the people will get too much confusion here. E alone it can be regulated by E. Which one? CDK2. CDK1 is B, but both by A. So now I am going to show you the question. Even though you know the answer, it doesn't matter to me. I want you people to spend 30 seconds on the given question. If you never improve your patience level at this particular moment, the day of examination, trust my words, I am telling with experience, you will get super excited. Because of your excitement, even though you know the answer, trust my words, you will end up by choosing the wrong answer. So friends, while answering the question, peace of mind is very important at this particular moment, because your exam is hardly 1 month is there. Okay, so please maintain your patience level. It's not about the right and wrong, it's only about how nicely you are understanding the question. If you answer the question with the patience, automatically you answer correctly. Okay, yes everyone, it's a part B question. Cyclin B assist in the activation of the CDK1 protein kinase at which stage of the cell cycle? Deepika, without fail watch the first half an hour recorded also. And Deepika, I want you to attend complete 30 days. Please make use of this session. Okay, yes. Okay, even though you know the answer, friends, take time, 30 seconds, slowly. Yes, yes. Neha said answer is 4. Okay, I told you na, the question seems to be very simple, but the question is very logical. Okay, others, friends, please listen. Right or wrong, everyone, please give me the answer. Everyone. Yes. Others. Only 3 people has given the answer. Can anyone give me the answer? Friends, such a easy question. Okay, the question says that cyclin B assist in the activation of CDK1 protein kinase at which stage of the cell cycle? Undoubtedly, friends, it is going to control very good G2 to M phase, because we have very clearly studied, right? G1 phase is under the control of cyclin D, cyclin E controlling S phase, cyclin A controlling S phase, completion of S phase. Very simple. Obviously, friends, cyclin B is the one which is going to control G2 to M phase. Wonderful, amazing, fantastic. Everyone has given the right answer. That's good. So be patient, be calm. Then automatically you can answer. Why I am struggling is this question is fix in your mind that you are going to get the 4 marks. Just fix in your mind blindly, so don't miss a small concept from this particular area. Okay, now let's start. 4 classes of cyclins are there, friends. We have discussed that. First one we can call G1 cyclin, another one we can call G1 to S cyclin. So please listen me for another 20 minutes continuously, then I'll make you solve the question. then S cyclin, then friends another one we can call it as the M cyclin. Please listen. G1 cyclin function is what? I am using a word that it will just start the G1 phase. Whereas G1 to S cyclins, what they will do, you know, everyone listen, G1 to S cyclins will helpful in ending of G1. They will helpful in ending of G1 and starting of S phase. Whereas S cyclins will be involved in the regulation of S phase. Whereas friends, what about the M cyclins? They are exclusively responsible for the process of controlling the mitosis or the cell division. Okay, and that point is very clear. But what we studied very clearly, we studied that the cyclins are having extremely low lifespan. Since the cyclins are having very low lifespan, the moment cyclins get activated, what it is going to do? They are immediately activating the CDK molecule. Now, entire cycle can be controlled majorly by the CDK. But the point is very clear, and this is the once again asked 4-marker question. The CDK activity can be regulated by two major mechanisms. One is what? CDK has to be regulated by the cyclin compulsory, and the second regulation that what we studied is phosphorylation and dephosphorylation of what? Key amino acid. This also you can get as a part B question. Which of the following amino acid involved in the phosphorylation and dephosphorylation of the CDK protein in the cell cycle? Like this also you can get the question in the part B. So what will be that? Will be the tyrosine. Please don't get confused with that. Now you listen, once again don't confuse. I have told to you that friends, if the CDK wants to be activated, inside the CDK, the tyrosine has to be phosphorylated, no doubt about it. And what is the goal of CDK? To the targeted protein, it is going to phosphorylate. Now, in the target protein, at which amino acid it is getting adding the phosphate group? CDK will add the phosphate group to which amino acid? That is the question. In the targeted protein, majorly for the serine and threonine. Don't get confused. CDK wants to be activated. Inside the CDK, tyrosine has to be phosphorylated. Once the CDK get activated, it is going to activate the targeted protein by adding the phosphate group majorly to the serine as well as the threonine. Please don't forget that particular statement. It's important one, friends. And you only remember the movement. Remember friends, the moment cyclins activate the CDK, what is happening? Cyclins are getting degraded very rapidly. So that's why we will use the statement that CDKs are inactive unless they are associated with a cyclin. But the cyclins are having a very less lifespan, friends. That's why cyclins turn over very rapidly. Now you can ask me a question, sir, how cyclins are getting degraded? That is an important question which is asked. Cyclins are getting degraded by an important protein complex. What is the name of that particular protein complex? We will be calling it as the proteasome. Proteasome is the name of the protein complex. All the students listen. Proteasome is the name of the protein complex which is degrading the cyclins, especially the cyclin which is present in the G1 phase. Okay, I want every student has to remember the name of the proteasome which is degrading the G1 cyclin. The name of the proteasome is SCF-E3 ubiquitin ligase is the name of the enzyme. Which enzyme it is? Anyway, I'll explain you. It is the name of the proteasome. What do you mean by the word proteasome? Proteasome is the degradative enzyme. Proteasome is the destructive enzyme. It is going to destruct the G1 cyclins. So now you can ask me, sir, the same enzyme Is it degrading G2 cyclin? Is it degrading the M cyclin? No, that is absolutely wrong statement. Every cyclin can be degraded by a specific proteasome. One proteasome cannot degrade every type of the cyclin. Different cyclins, different proteasomes. Since we are talking about the G1 cyclin, that can be destructed by this particular enzyme. Okay, now all the students look into this diagram very carefully. A very important diagram. Look into this carefully and you have to answer, give the answer correctly. What is the statement I have given to all of you? If the CDK molecule wants to be active, it is compulsory that it has to be it has to be associated with the cyclin also, and it has to be associated with the phosphate group also compulsory. That is what I told to you. You only look into the picture, every student. This is the CDK molecule. How many phosphates are there? Two are there. So but phosphates are associated with the tyrosine only. Suppose see here, this is we can call inhibitory phosphate and this is called as a stimulatory phosphate. Question is very clear. All the students listen. I am calling this CDK as the active CDK. Why? Why I am calling it as an active CDK? I called it as an active CDK because the first reason, it is associated with the cyclin, the first reason. Second reason, it is also phosphorylated. Which tyrosine get phosphorylated? That's the valid question. Stimulatory tyrosine get phosphorylated. That's the reason I am calling it as the active CDK. But in the second case, why did I call it inactive? Of course, cyclin is associated. Then I should say active, right? No, I never. Because the stimulatory tyrosine is not activated. Hence, I never say inactive, right? And friends, you only look into the picture. The cyclin activated the CDK molecule, and what did I tell you, friends? The cyclin having a very less lifespan. Every student please look into the diagram. This cyclin is associated with what, friends? The cyclin is associated with a proteasome complex. See here. Now everyone has to remember my words. Now you have to get a valid doubt. Sir, this is a very valid doubt. Sir, how does this proteasome enzyme comes to know where is the cyclin? How it exactly comes and degrade the cyclin very accurately? That is a very valid point. Because friends, a proteasome can't detect any protein. But what is the statement you have to remember? The cyclin, whenever it wants to be degraded, it is added by the ubiquitin molecule. You have to remember that. Keep it in your mind. Whichever the molecule that is associated with the ubiquitin, that ubiquitin associated molecule can be easily degraded by this particular proteasome. That's why we can call it ubiquitin mediated proteasomal degradation. What do we call, friends? We call ubiquitin mediated proteasomal degradation. Please don't get confused with that particular point, right? So after that, cyclins get degraded, undoubtedly CDK will become inactive. Okay, now don't be hurry, think slowly. Even though you know the answer, read the question and then only give me the answer. Yes, everyone. Take time, don't be hurry. Friends, even though you know the answer, don't be hurry, please. Neha given 3, Sneha given 3, Sonam 3. Others. Shivani also C, Priya C. Others. Okay, let's look into the given question correctly. Okay, cyclin dependent kinases which control the progression through cell cycle checkpoints are fully activated by which of the following? Friends, this is the statement you people has to remember. They have given a word that fully activated. So when it get fully activated, if only cyclin, it never fully activated. If only CDK get phosphorylated, it never activated. So both the cyclin plus phosphorylation by a CDK activating protein kinase, then only it will be active. Only phosphorylation is not the answer. Hence the correct answer will be 3. No, Apurva, it cannot be B, dear, because we discussed it, right? You will look into this. Cyclin has to be compulsory associated with both with the CDK molecule, and it has to be phosphorylated. So it never be a single mechanism. Undoubtedly it will be a 2 mechanisms. Correct, na? So the correct answer will be obviously C. Okay, perfect. Okay, that's great. Okay, so friends, now so far what we have studied? So far we have studied what are the different type of checkpoints are there, what are the different functions of the checkpoints, and what kind of CDKs and what kind of cyclins are involved. So now, because it's a broad concept, now we are going to discuss about every checkpoint very briefly and elaboratively. So before explaining the individual checkpoint, I want you to answer this question. This question is a very tricky question. Don't be hurry. Don't give the answer, with anxiety. Think and give me the answer. Simple question but makes you confused. It's a simple question but gives you with confusion. Think the passage of cell. It's a part B asked question. Yes, what is the answer? Right or wrong, no problem, tell me. But anyway, you have to give me the right answer only. Think it. See, don't think too much. The best way to give the answer is making it simple. Just make it simple, that's all. Automatically you can answer. Sneha said B. Just make it simple, friends. Don't think too much. And it's a very simple question. The question says that the passage of cell through the stages of the cell cycle is controlled by a protein kinases that phosphorylate many different appropriate protein. What is the name of that kinase they have asked? Of course, they are the CDKs. Correct, na? What I told to all of you, friends, CDK is going to phosphorylate the targeted proteins, especially where it is going to target serine and threonine. No, smart, that is not the answer. It cannot be a tyrosine kinases. The answer will be the CDKs. They will majorly pass for if they ask, because if they ask CDKs act on the targeted proteins, specifically on which amino acid, that kind of questions may expect. So you have to remember very specifically on serine as well as the threonine. Don't get confused with that. Okay, now what we do is We have understand different type of CDKs and different type of cyclins involved in the every phase. We also studied very clearly I mean to say at which phase what kind of CDK and what kind of cyclins are involved. Now we have to study very depth. Now we have to understand how does the G1 checkpoint works, how does the G2 checkpoint works, how does the M checkpoint works, and most importantly, how these checkpoints are getting regulated. So once we understand this particular checkpoint regulation, the concept is crystal clear. Okay, pay attention and please listen to me correctly and answer the questions correctly. If you listen me correctly, you will answer correctly. G1 checkpoint. Friends, the moment you heard the name of a G1 checkpoint, the first point has to come in your mind. What the G1 phase is going to do. Keep it in your mind. G1 checkpoint is going to take a decision. It is going to take a decision. What decision it is going to take? Whether it has to yellow the cell to enter into the S phase, it has to yellow the cell for the division or not. That is getting decided in the G1 checkpoint only. In case, every student listen, you will automatically understand. In certain cases, suppose the cell is not properly prepared for the S phase. Once again listen, the cell is not properly prepared for the S phase. Then what is happening? The G1 checkpoint will stop that cell and it will take the cell to the G0 phase. You know, right? G0 phase we can call the resting phase, and widely we can call, my friends, a quiescent phase. What do you mean by the word quiescent phase? 90% of the students will think that quiescent phase means it is inactive phase. See, listen, the word inactive is correct. It is only inactive for the division, that's all. The cell is not dividing, that's all. But my friends, the cell is metabolically active. You have to remember the statement. If the cell is not metabolically active, friends, the cell may die. So never confuse that G0 phase means the cell is getting inactive. That is true statement. But the cell is inactive only for the division. In terms of metabolism, the cell is extremely active. Many cells stop at this stage and enter a resting state called as G0. And I want you to remember these two statements. Liver cells as well as brain cells, they only enter the mitosis around once or twice in a year. That's all. Then remaining all the time, especially the liver cells, throughout the year, they will be in the G0 phase only. Please remember that. And keep it in your mind, especially in our body, in the human cells or in the animal cell, the G1 checkpoint, widely called it as, friends, please pay attention, we can call it as the restriction point. What it is restricting? It is restricting the cell and deciding whether the cell has to enter into the S phase or not. So because, friends, if the cell is not properly prepared, and most important, if the cell is get damaged, how does the damaged DNA can undergo the replication? If a damaged DNA undergo the DNA replication, it leads to the mutation and it causes permanent, it causes cancers. So we have to block this. Now, see, friends, we are talking about the first checkpoint only, G1. The concept is a very broad concept. Have patience. Now, keep it in your mind, friends, G1 checkpoint will do two major functions. Everyone remember, G1 checkpoint will do the two major functions. What is the first function? G1 checkpoint, it is having two sub checkpoints. What is that? One checkpoint we can call the damage checkpoint. What do you mean by that word? Which means this checkpoint only check is there any damage in the DNA. That is the only function it will do. Then second one, friends, we can call DNA replication checkpoint. What do you mean by that? Which means the second point is not at all concerned anything about the damage. It can't do anything if the damage occurred. The second point is only checking whether the DNA replication occurring with a great accuracy or not. That is the only function of the second checkpoint. So keep it in your mind. Keep it in your mind. G1 checkpoint function is to take the decision to allow the cell for replication and division or not. That G1 checkpoint contain further two sub checkpoint. One we can call damage checkpoint, another one we can call DNA replication checkpoint. One checkpoint can only check the damage and try to resolve that. Another checkpoint only will check whether the replication is occurring properly or not. That's the only thing it will check. Now let's talk about this. So all the students has to listen to me very carefully, friends. This is a very important one, because 90% of the time we can get the questions in the form of statements. In order to answer that statement, you have to know the concept correctly, because all the statements are more appropriate, very tricky. See friends, here there is a double strand break occur. The reason may be anything. The reason may be UV radiation. The reason may be treating with a chemical agent. The reason may be anything. There is a double stranded break occur, or the DNA replication gets stopped at the replication fork. Whatever the reason, immediately what is happening? You people should not think that whenever the damage occur, the cell will die immediately. That is completely a false assumption. The moment the double-stranded DNA breakage occur, the cell never die. First, immediately, the first event that has to be happened, because friends, the question come in this way also. What is the first event that occurs after the damage? Once the DNA get damaged, the first event that takes place is the cell cycle get arrested. First one, cell cycle get arrested. And second one, what is happening? Immediately friends, the checkpoint only activates. What it is activating? It is activating several DNA repair mechanism and respective enzymes. That repair mechanisms and enzymes will try to repair that damage. In worst cases, if they are unable to repair that damage, what friends, if they are unable to repair that damage, only during that particular condition friends, the process of apoptosis will take place. That is the programmed cell death. So now you can ask me, sir, why the apoptosis has to occur? Is it a necessary process? It is mandatory process. The reason, the cell got damaged. Now you only understand, if the damaged DNA undergo the replication, if the damaged DNA undergo the division, it leads to the cancers very fast. So we should not keep that cell as it is. We have to kill that particular cell, then apoptosis occurs. So what is the statement I would like to conclude? Apoptosis is the final step. Apoptosis is not the first step. That is the point you have to remember. Whenever the damage occurs, the first key event that happens is immediately checkpoints will arrest the cell cycle. They never allow the cell to move. Then they immediately activate the DNA repair mechanism and they will try to repair. If not, they will kill the cells. So, but they are finally killing the cell, but they make sure that whatever the cell got damaged, it never allow into the next stage. That is the unique statement. Now listen. So let's talk about the two things. What did I tell to all of you? Please don't forget friends, remembering is very important. Damage checkpoint, DNA replication checkpoint. Correct na? Now let's look into the DNA damage checkpoint, which is the sub checkpoint of G1 checkpoint. I told you, right? G1 checkpoint having two sub checkpoints. First checkpoint we can call the damage checkpoint. Now here comes a very interesting point. What is that very interesting point? Now you have to ask me, sir, how does checkpoint comes to know? Every student is listen. How does checkpoint comes to know the cell is, the DNA is damaged or not? That is a valid question. How the checkpoint comes to know? Every student keep it in your mind. The checkpoint, this is a very interesting point. The checkpoint contains sensors. The checkpoint contains sensors. What the sensors are doing friends? The name itself indicating the sensors, which sense something. What they will sense? They will sense the damage. That is how the checkpoint recognize the damage. Every student compulsory remember the name of the sensor, that is called as ATM ATR. You no need to remember the full form, it's not necessary. ATM and ATR are the type of kinases that are present in our body which check the damage. The same ATM and ATR, they are present in the yeast also, which is undergoing the budding. That is called TEL1 MEC1. They are also sensors present in the budding yeast. TEL1 RAD3, they are also sensor kinases present in the yeast which are undergoing the fission. If you can't remember the sensor kinases which are present in the yeast, no problem at all. Remember the name of the sensor kinases which are exclusively present in our body, ATM and ATR kinases. Very simple, right? Now, okay, we have recognized the damage. After recognizing the damage, what we have to do? We have to stimulate lot of enzymes. What enzymes? What enzymes has to be stimulated? Friends, DNA repair enzymes has to be stimulated. You have to understand me correctly. Whenever ATM and ATR, they are what? Kinases. ATM and ATR what, friends? Kinases. Once they sense the damage, they will activate the DNA repair enzymes. The question is very clear. How they are going to activate the DNA repair enzymes? By the process of phosphorylation. Very simple point. Now you have to ask me one more doubt. Sir, does the DNA repair enzymes present in our body, will they be not active all the time? No. DNA repair mechanism enzymes all the time, they never be active. They will be active whenever there is a damage occur. So someone has to recognize the damage. So who is going to recognize the damage? ATM or ATR recognizing the damage, activating the repair enzymes by the process of phosphorylation. Then the enzymes will come into the picture. Then they will do the repair. Please don't forget. Okay, so please remember, friends, ATM and ATR, these two are the major kinases which are present in the G1 checkpoint, acting as a sensors. Acting as the sensors. I am requesting all of you, friends, please remember ATM and ATR, please remember, belongs to the family called as PIKK. I want you to remember this. They are belongs to the family called as phosphatidyl inositol kinase like kinase. What do you call it as? Phosphatidyl inositol kinase like kinase. This is the name of the family. Don't forget. Now you have to remember what kind of damages can be recognized by the ATM. Now the question is very valid. Do you think ATM is going to recognize every damage? That is wrong statement. ATM exclusively recognize only double-stranded DNA breaks. DSBS, double-stranded DNA breaks, can be specifically recognized by the ATM. When it comes to the ATR, ATR can also recognize the double-stranded break. ATR can also recognize different DNA lesions like deletion, like duplication, like inversion, like translocation. Even, friends, whatever the errors that are encountering in the replication, entire errors can be recognized by a sensor kinase, we can call it as ATR. So ATM and ATR are the two major type of sensor kinases which are belongs to the family called as a PIKK, that are involved in recognizing all the damages. Never ever forget that. Okay, that is what, friends, I told to all of you. What did I tell you? This is a damage checkpoint. They will recognize the damage, activate the DNA repair mechanism enzymes, they will repair it. That's all. Now, what is the second sub checkpoint of the G1 checkpoint? This is DNA replication checkpoint. This is also very accurate one. Now, all of you answer. Does the DNA replication checkpoint, do you think it can recognize any double-stranded breakage? Does it recognize any lesions that are occurred in the DNA? Never. It never involved in that. This DNA replication checkpoint is exclusively responsible only understand is there any error is occurred while replication is occurring. Because friends, I have told to all of you multiple times, our DNA do not contain one or two nucleotides. Billions of DNA bases are there. I told you many times, if you keep the DNA on the floor from planet Earth, it will go to planet Mars 600 times. That much long molecule DNA is. So which means billions and billions of nucleotides has to be replicated. We all people studied in the molecular biology also very clearly that our DNA polymerase enzyme will make only one mistake, just one mistake out of 10 lakhs of nucleotide added. Out of 10 lakhs of nucleotide added, only one mistake happened. But the question is very clear, my friends, even though one mistake occur, if we don't resolve that mistake, if that mistake occurred in the coding region, which leads to a great damage, which causes many genetic abnormalities, many. That's why what this replication checkpoint will do, it makes sure that the entire bases will occur or will undergo the replication very accurately. Okay, now a question is very clear. What is the name of that particular protein that is controlling the DNA replication? The name of the protein we can call the PRB protein. This is the question which is asked in the 2022 February exam, which happened 5 months ago for the part C section, direct part C question. Now you all people know that RB gene, it is the major gene coded by what? This is the RB gene which is coding for a protein called as PRB protein, which means protein retinoblastoma. What friends? Protein retinoblastoma. Please don't get confused with this. And please remember, this is the first identified tumor suppressor gene. Friends, you all people know, right? There are many tumor suppressor genes are there. There is a P53 gene is there. There is a P21 gene is there. There is a P16 gene is there. There is a PRB gene is there. Most of the students will get confused between all these. PRB is the first identified tumor suppressor gene. First identified. But the most widely studied tumor suppressor gene in the cancer biology is the P53. Most studied. We will read in the cancer biology chapter, don't worry at it. So PRB is the major protein which is going to control the G1 to S phase transition. I can tell you very clearly, PRB is going to control all the events that are occurring in between the G1 to S phase. Okay. So friends, how does this PRB will act? That we will discuss in the tomorrow class. Okay. So in the tomorrow class, we will complete the G1 checkpoint, G2 checkpoint and M checkpoint. Okay. And listen everyone. If you have attended today's class and if you don't attend tomorrow's class, which is the continuation of this, trust my words, you don't get the concept. Once I explain the entire concept, then only you will get a crystal clear idea. Okay. I am reminding you once again, my goal of this 30 days is to give you the correct knowledge about that particular topic and solving the part B and part C question. My only request from all of you is till September 13th, please attend regularly and have good patience and interest. That's all I'm expecting. Okay. But it's my responsibility. I'm telling you, if you attend these 45 topics, I am assuming you will get more than 100 marks for sure. Very, very sure. Okay. And friends, the notes cannot be posted in the Telegram channels. You have to download my app. The app named as Chandu Biology Classes. The link is there in the Telegram, or you can directly type in the Play Store also, Chandu Biology Classes. Okay. So if you download today, by tomorrow afternoon, once I finish the entire cell cycle regulation, then I will add the notes in that particular section. Okay. Along with the part B and part C questions. From there you can access. Okay. Okay friends, I hope the session is useful to all of you. Please respond, say yes or no. Is it clear everyone, the lesson? Please respond. Is it clear? Yes, perfect. Okay. Okay. Thank you all of you. I'll see you in the tomorrow sharp 3:30 p.m. Okay. Thank you all of you. You can ask me any kind of a doubt. Okay. Apurva, in the description only you are having the Telegram link. You can see it. Okay. Okay friends, thank you all. I'm requesting all of you, don't waste the time. Keep on working and keep on reading. Don't waste the time. Thank you all.